Hi, my name is Katie Hooker. I'm a molecular and cell biology major from Guilford, Connecticut, and I spent the summer researching the relationship between a particular gene called FOXP2, the brain, and reading and language skills. So reading is a critical milestone in child development that's a necessary skill for most other learning endeavors. And what I mean by this is it's the foundation for a lot of other academic domains. If you can't read, you can't do science, math, history, and English. And therefore it becomes increasingly difficult to progress academically. And because reading is such a necessary skill for our daily life, individual differences in reading and reading disability are an important area for further study. And in working to understand the mechanisms underlying reading and reading-related skill, we can hopefully work toward earlier identification, treatment, and more efficient intervention for children with reading issues. So reading depends on various aspects of language, such as meaning and sound structure, as well as some general cognitive skills like working memory and attention. And we also know that reading disabilities like dyslexia run in families, thus demonstrating a genetic component. Although we're aware that this genetic component exists, we don't fully understand these complex links between particular genes and reading traits. So our problem is that our current research really only accounts for a very small % of the variability we see in the general population as it relates to reading and reading-related skill, and this study works to contribute to this set of understanding through a gene called FOXP2. So the next important factor I'd like to talk about is that we know reading and language depend on each other. We know, based on previous study, that regions of the brain that subserve language also subserve reading, and we can therefore say that they're connected and not separate traits, and it's reasonable to examine them together, as I do in this study. So I've mentioned FOXP2 a couple times, and I just want to provide a little bit of background information as it proves to be a critical turning point for the duration of the presentation. It's of relevant clinical interest because of the severe effects observed in the family in which it was discovered, and this gene is extremely important for our understanding of speech and language. So most of our current understanding to date of the FOXP2 gene comes from the KE family and a hereditary speech and language disorder. And affected members of this family had a severe impairment in speech production as well as certain aspects of receptive language. And this study concluded that one mutated copy of the FOXP2 gene leads to issues sequencing facial muscles in order to produce proper speech. The gene also encodes for a protein of the same name, and this protein interacts with other genes. So a minor change in the FOXP2 gene can lead to alterations in expression of multiple genes, which can have potentially cascading effects. And these changes or variants in the genetic code can be studied through human genotyping and linked to diseases and behavioral phenotypes as done in this presentation. And the link between FOXP2 genetic variants and phenotypic deficits in the general population is largely unknown. Therefore, it's necessary to direct our research to common FOXP2 variants in the whole population in order to understand its potential broader impacts. So since we know reading is built upon language, and we know that FOXP2 is important for language, it's possible that reading is also affected by variation on the FOXP2 gene. So the approach for this study basically works to understand the relationship between gene, brain, and behavior through combining molecular, genetic, behavioral, and neuroimaging analyses. So genes are related to behavioral traits through mechanisms at the brain level, like processes in brain development, and these processes in turn determine the structure of the brain. So for these analyses, I first examined the relationship between the FOXP2 gene and behavioral measures, and then I followed this up with an anatomical brain analysis in order to enhance this understanding. So at the beginning of the summer, with the help of my mentors, I set out on 3 goals. These were to first identify an association between FOXP2 and expressive language in the sample of interest, because we already know FOXP2 is implicated in oral language measures. To explore an association between FOXP2 variants and reading in order to improve our understanding of these biological mechanisms, and to examine a potential association between FOXP2 variants and brain structures implicated in reading in order to enhance our behavioral understanding. So the next important factor of my project are what are called single nucleotide polymorphisms, or SNPs. And these are basically single base pair substitutions that commonly vary across a population. So SNPs are found within our genes, within our DNA, and they're in the actual base pairs themselves. And so although these changes may seem small, they can have cascading effects on how a gene, protein, or even individual functions. So for the purposes of this study, we considered two groups: people who were homozygous for the more common or major allele, and therefore had two copies of this more common allele, and people who were minor allele carriers and had at least one copy of the less common allele. And this was done for each particular SNP examined. So at the end of the day, our individual differences really do boil down to these small, fine-grained genetic differences like SNPs, despite the common pop cultural belief that large-scale mutations make us unique. And if we didn't all have these small differences, we would all essentially look and act the same. So for the purposes of my particular study, I looked at 4 SNPs on the FOXP2 gene that have been examined in previous studies of a similar nature. So the oral language measures I examined in my analyses were oral expression, articulation, and phonological awareness. Oral expression is a measure of expressive language skills, and it contains 2 subscores, picture vocabulary and story recall, which were included in the analyses separately as well. Articulation is a measure of production of speech sounds, and phonological awareness is a measure of a child's ability to manipulate speech sounds. And then in terms of reading, it was tested at 2 levels: word level and passage level. For the brain analyses, the inferior frontal gyrus, precentral gyrus, and basal ganglia were examined in both hemispheres of the brain, although it is important to note that we typically see associations with reading and language skill in the left hemisphere of the brain. And these regions of the brain have all previously been associated with speech production, including the control of articulatory muscles, and have been examined in studies extensively involving the KE family. So where is this data coming from? I used a pre-existing data set that has already been used in a previous publication of a gene called BDNF. And more specifically, after subsetting this data set, I found 115 participants available with available FOXP2 genotyping data at all 4 SNPs of interest. And these participants were children who had completed a battery of neurobiological and behavioral assessments in language and reading, and on average were between the ages of 6 and 10. So for the purposes of my project, 2 of my SNPs were found to be frequently inherited together, and this is what is known as linkage disequilibrium. And basically what this means is inheriting one version of one SNP often allowed you to predict what version of the other SNP they inherited, so these analyses could be collapsed into one analysis as opposed to 2 separate. And without getting too much into the details, the behavioral analyses were essentially a statistical test that allows you to compare groups while controlling for an additional variable. And this allows you to make sure that the differences between groups you observe are attributed to the variable of interest, in this case genotype, and not any additional variable. And this is called an ANCOVA. And these analyses tested for significant group differences between genotype groups at each SNP for each behavioral measure of interest. The brain analyses, however, were very similar to the behavior, but a SNP was only included in the brain analyses if it showed significant results in the behavioral portion. And this analysis used ANCOVAs to test for group differences between genotype groups on measures of surface area, thickness, and volume, which are features of brain structure that have previously been associated with language-related traits. So one of the tested SNPs did show a significant group difference for the story recall test. So this means that individuals homozygous for the more common allele at this SNP performed significantly better than children who carried the minor allele, and the story recall test involved repeating stories verbally. In terms of the brain, none of the selected regions of interest showed significant group differences on measures of surface area, thickness, or volume. So this study was designed to examine group differences in a variety of reading language and brain metrics across various FOXP2 variants, and we observed a significant difference between genotype groups for one SNP for the story recall assessment. It's important to note that both groups did in fact perform above average, as the mean score for this test was 100, but it seems that the lower performers were captured in that minor allele carrier group. And this discovery aligns with the context of the FOXP2 gene and its implication in oral language and articulation, and it supports the initial hypothesis that major allele homozygotes would outperform the minor allele carriers on any given behavioral test. In acknowledging the limitations, the population that I examined was typically developing and therefore not representative of poor reading ability or disability, so the group differences in reading ability seen in this population were rather small. So it's possible that FOXP2 might play a role in some of the larger differences in reading skill that were not represented in this population. In addition to this, the lack of statistically significant differences observed at both the brain and behavioral levels could also be due to this sample composition. And if a sample including children with reading and language difficulties were to be examined, there may be stronger effects at both the behavioral and brain levels. And this is also further supported by our current understanding of the FOXP2 gene, as the population in which it was discovered presented a rather severe phenotype. And lastly, the number of subjects included in the brain analyses was much smaller than that of the behavioral analysis, simply due to a lack of valid MRI data. And even within the context of genetic studies, the sample size for the behavioral analysis was rather small. And this is particularly important to acknowledge because the study focused on SNPs, or relatively small changes in the DNA, so a larger sample might be necessary in order to see statistical significance. So in the future, this study should be replicated in another larger, more diverse sample, which is something I might be working on to come. So that's all from me, but I just wanted to take a moment to thank my mentors, Megan Perdue and Dr. Landi, as well as the Holster Scholars Program, the UConn Honors Program, and Mr. and Mrs. Holster. Thank you.